I am Professor S. Jeevan Chandra, Chaitanya Deemed University. Today, I would like to discuss about the immune evasion mechanisms of Omicron variant. So, what do you mean by immune evasion mechanisms? Immune evasion mechanisms are the mechanisms ex- exhibited by a virus which masking the attack of T cell receptors to mold specific antibody production. So what happened? They are escaping from the attacking T cells, thereby they do not offer the production of neutralizing antibodies to particular viruses. This leads to complicated procedures. That means the viral variants could not be neutralized by the existing antibodies. Coming to this system of immune evasion was first recognized itself by the first variant of Wuhan city, that was alpha variant beta 1.1.7. This is the alpha variety. In the alpha variety, you see here the genome of the entire system is very characteristic and it has very important sites. This is called a receptor domain, and it is exactly about more than 1200 bp of the bases of the RNA, and exactly at 700 region there is a furin cleavage site, and from 300 to 500 is the receptor binding domain. But majority of the mutations which will occur in particular type is depends upon the genome integrity. The virus is itself is a RNA. So what happens? RNA is less stable and it is utilizing the host epigenomics properties like methylation, 7 methyl guanine, 5 prime capping, 3 prime adenylation, and special systems can be utilized. So it is a animal virus. When it has entered in viral cell through endocytosis, now immediately the viral genome get 7 methyl guanylated and it will protecting from the attack of RNases, restriction enzymes. So RNases are there, they are the surviving factor for us, and they will control the invading RNAs. So now alpha variety was changed into beta variety, that is beta 1 comma point 351. The 351 site is getting converted, then beta converted into gamma at P1 location, exactly 471 484 sites. Then it was leads to formation of delta variant beta 1.617. This 617 site has converted into a mutation called D950 also. So these all are the mutations, these hotspots, rings that frequently undergoes mutation, which leads to new variety of systems. Coming to Omicron case, Omicron has more than 50 types of mutations for each 100 amino acid residues. For example, ranging from A amino acid 67 region, G 142, 211, 390, 417, 547, 614, 764 and 865, 954 histidine sites. These all are the most important things. But here the 3 sites are very important at the 471 to 529 sites. These all are the very common things. These all are contributing for receptor binding domain changing, as well as the coming to modification of the receptor itself and spike proteins itself. So both are duly affecting the system. This is the case how it leads to formation of new variety in that particular system. This first variant alpha, then followed by beta, gamma and delta. Now a new variety was changed into a variant called beta 1.1.529 is called Omicron on 24th November of 2021 in South Africa. What is the background? The first confirmed beta 1.1.529 infection is from a specimen collected in November 2021. The cases of this variant appears to be increasing in almost all countries, and variant has been detected faster rate than the previous infections. Why? Because the mutated regions at beta 1.1.529 mutation profile it several binding domains. That means it is distant location, but that means not at the 100, 200, 300, 400, 500 sites. It is especially at the distant location of the genome is ranging from 800 to 900 region, where it is changed the delta configuration. Now it leads to Omicron variety. Coming to sites, especially the variant with documented with different phenotypic effects and genotypic insertion deletions, and which will promote the amino acid configurations of the system. Coming to this system, the Omicron variety has accumulated the 79% of infection rate even in vaccinated individuals, and 16% of infection rate in reinfected individuals, and most cases are going to be occur between the age group of 18 to 39 only, but above 40 and below 18 there are no cases. This tell us the story how youngsters are neglecting their daily life and daily events. Coming to variations and mutations, how basis of the COVID-19. So COVID-19 is a RNA virus. So this RNA virus generally adopting the RNA protection activity. The RNA, how best it could be protecting? As I said that either modifying the 5' site, 3' site. 5' site is 7-methyl guanylated and other sites also methylated, and 3' region is polyadenylated. So these all are nothing but mutations or sudden inheritable changes that will occur in the host cell itself. Within the host cells, all viruses of COVID-19 have been mutated at their possible rate in their genetic code. Most of the mutations are occur in spike protein. This spike protein is S protein, which contains a very dangerous activity, that one in beta and alpha and gamma and delta, and even in delta up to delta, D641 followed by G. That means amino acid D aspartate was changed to glycine, that was the delta case. Now this 641 of the viral spike protein very stable in alpha, beta, gamma and delta. Now it has changed to another facet called Omicron. So not only that, this leads to changed spike protein. The spike protein will lower the affinity towards gateway of ACE2 receptor. The ACE2 receptors are found on all what you call all nucleated cells. So the ACE2 receptor is going to be not identifying and properly molded the response. Next, what happens? The Omicron variant has 43 amino acids residues are getting mutated in the spike protein, from which 34 residues will contributing for 60% of all available sequences will show various types of mutations that will occur in different sites of the spike protein. You see here there are different types of mutations are there. The rate of mutations are more in the 350 to 550 region. That means 529 is the region, that means Omicron was produced. These domain, so from 377 to 529 and 530, glycine at 446 and 440, these all are the sites, the red color sites are frequently changing. So due to changing of the key, what happened? Lock was not properly open up. Same manner, the Omicron also contributing for these aspects. Coming to the nature, now mutating virus, Omicron has more mutations that leads to more variations of the spike. So when you compare the spike of Delta at B617, where the mutations are going to be changing, and that Omicron has more than 70% similarity with Delta, 30% is different from the Omicron. This change will contributing for more stability of the virus, and it will be going to be leading to changes. 32 out of 50 mutations of Omicron are just occurred on spike. Spike is the red color region. This orange color region is nothing but spike present on the virus. This is the spike structures. Most of them are occurred at the 529 to 377 region. So this is the leading to contribution for different types of infection rate. So red color regions will contribute by more than 70% infection rate, and yellow colored regions will contribute 15 to 40% region, and green color will be 5 to 15%, but blue color one 1%. That means these are the sites which are sequences derived from the patients. So they will show how lower affinity of the spike was contributed for the system. And coming to the virus, what happens? The high number of mutations will does not automatically meant for the variations, but they are also more dangerous. That's why virus is changing its face. One person will infecting with one variant, he delivering another variant. Like that, this variant is going to be changing. And antibodies, most of the antibodies acquired previous through the infection or vaccination or recognize the spike proteins and prevent it from the attacking the host generally. But what happens due to changing the spike configuration, now the any change in the spike can potentially affect the virus identification and virus neutralization. Now the virus capsid, nucleocapsids are also changing their genetic code. These are contributing for change on the spike, that potentially changing the identification of the virus. So that's why virus is going to be changing its the it conditions. So coming to the why spike is a matter of identification, because as we have previously discussed that the spike protein is a key protein that going to enter and interacting with the receptor of the ACU2 receptor. The sites present on the SARS-CoV spike, red color are more than 70%, 1 to 50% green color. These all are contributing for more dangerous infection rate and fatality of the infections. So what happened in the first cases of South Africa and over the world, 24% are fully vaccinated, but they are getting infected. Of them, so 75% of new cases in the country. That means those who are not infected are getting affected by this virus. And finally, after 40 days vaccination, all countries, 11 countries are affected with Omicron so far. And now the Omicron was spread to all over the globe, including 40 to 50 countries, and with a fast infection rate. So this tells us the importance of Omicron in contributing for the immune evasion mechanisms. Let us discuss more about immune evasion mechanisms. What made this virus to be evasion from the immune system? Why it should evade from the immune system? Immune system is a policeman which recognize the new coming variant, but due to changing of its face, the spike proteins, the lock and key fit molecules, that means spike is the lock, receptor is the key, both are not configured perfectly, so they are escaping from the attacking APC cells and T cells and B cells. So T cell receptor is unable to generate epitopes for B cell recruitment and antibody production. Now the antibody production was not there. And another more important thing is the shortation of the incubation period. Previously it was from 1 to 15 days in the alpha, then beta variety 1 to 7 days, in delta variety 1 to 5 days. Now it has been decreased to 1 to 4 and 3 days. Within 3 days it will be replicating more faster. After 5 days the virus will be excluding from your body with a sputum and something. Within 7 days of period it will be slow down. But how it is going to be escaping from the T cells? Because it is masking itself and the mutations are contributing for this variety. So whether it is fatal one, in some immunocompromised individuals it is very fatal, but in immunized individuals it won't affect you. So booster dosed individuals and 2 dosed individuals are highly protected, but unvaccinated individuals are suffering a lot. Another more important thing is that generally our immune system is very alert and it will utilize settled kind of natural immune mechanisms to clear the viruses from your body. But existing antibody profile, convalescent seras does not confer the immunization. That means they are escaping from the antibody neutralization. That is a big problem. So how to overcome this? Because again we require specific vaccination development for especially spike targeted vaccinations. That means for each variety we have to develop a new vaccine, then only it will be protected. Simple thing, avoidance, personal hygienity and other personal protective measures will only protect the individuals, not the vaccine itself. But there is no prominent what you call treatment system in this process. Thank you very much for listening.